Well, thank you very much. It's an honor to be here, to be able to talk about what Frank described correctly as the coolest biology I've gotten to work on in my entire career. And the story really begins with this woman. This is Jean-Louise Calment. She's a resident, or was a resident of Arles, and very much a French woman. For her life, until she was 100, she rode her bicycle every day. She smoked cigarettes until she was 117. And her family refused to put her in a rest home up and until the point of just a few years before her death, when she somehow managed to light her kitchen on fire. Now, this picture, taken a year before her death at 122, Jean was the oldest living human being in history. Now, some demography published actually just 10 days ago suggests that the 150 years we've had of ever-increasing maximum and median human lifespan, that this increase has ground to a halt. And what we're observing now is the maximal, achievable, natural human lifespan. Now, I did not come here today to talk about natural human lifespan. Rather, I want to talk about augmented human lifespan. Put another way, what becomes possible if you're willing to disturb the universe of naturally occurring human biology? Well, if nothing else, I hope that if you guys are awakened 2 weeks from now in the middle of the night and asked to recall what it was I talked about, my hope is that you'll remember 3 core ideas. And the first is that aging is not a rigid, inflexible thing. Quite the contrary, it's a flexible, malleable thing. And nature has bent and twisted aging throughout eons of evolution to change the duration of time creatures exist. Now, to make that possible, nature has had to build a series of molecular control knobs. These are things which nature actually twists and turns to change the duration that a creature can exist. And then finally, we as scientists and drug discovery explorers are for the first time finding ways to turn these molecular knobs safely. What this means for all of us in this room is we're going to be entering an era in which there are drugs, safe drugs, that change the rate at which we age. Now, why do I believe all of this? Well, let's begin with aging as a flexible thing. So if you look across phylogenetic space, look through evolutionary history, and you look just in the class of mammals of which we are members, and you take the shortest living mammal, the tiny shrew, and the longest living mammal, the bowhead whale, no one knows actually how long one of these creatures can live. The best estimate is at least 200. And what you see is in a group of creatures, that is mammals, which are more or less identical in their biochemistry, you have creatures that have a greater than 200-fold difference in their duration of time they can stay alive. Now, if you look at a more closely related group, specifically the rodents, and you look at the field mouse, and you look at one of its closely related family members, the naked mole rat lives 10 times longer, and Brandt's bat lives as much as 12 times longer. And these are creatures not only with identical biochemistry, but that have been quite clustered in terms of their evolutionary history. Similar creatures, very different lifespans. If you go over to far away from mammals, go all the way over to mollusks, like the hard clam that's something people eat at a clam bake, can live a maximum of approximately 40 years. Well, its deep ocean-dwelling relative, the quahog, this thing, as far as people know, can live at least 500 years. Again, they live so long, no one really knows how long these can live. It's at least a 12-fold difference in maximal possible lifespan. So this tells you that nature has bent and twisted biology using something. And what it's been twisting has been these molecular control knobs. Now, scientists have recently figured out what some of these knobs are and have begun twisting them. So Robert Rice was able to extend the lifespan of a worm tenfold by modifying one gene. Valter Longo, modifying 2 genes and reducing calorie intake, gets tenfold extension in yeast. Now, a little closer to home, take a fly. Deleting 1 copy of a single gene doubles a fly's lifespan. And now let's get really close to home, a mouse, a creature that, while certainly so much smaller and less socially interesting than ourselves, it's a creature with the same biochemistry, mostly, that we have. And modifying a single gene and reducing calorie intake, you get a 2x increase in lifespan. Think about that for yourself. Imagine being able to live to 160 or 170. These are tiny changes. Now, this suggests that these knobs exist, but can we really turn them in a safe way? So the answer is yes. The first example historically of such knob turning by scientists was an accident. So it was in the 1930s, and a scientist named Clive McKay working at Cornell during the Great Depression. So one of the things about the Great Depression that they didn't have was money. And so he couldn't keep his colony of rats alive. So what he had to do was, in order to keep this precious colony alive, is he realized he had to feed some of them every other day. And some of them he fed every day. Something very surprising happened. The ones he fed every other day, they lived longer. In fact, as much as 30% longer. And it was the first time human scientists had ever figured out this was a knob you could turn pretty simply. Now, flash forward to the 1970s. This picture is, in fact, what you think it is. These animals are literally joined at the hip surgically. Their bodies have been fused. One is old, one is young. So the blood of the young flows into the old animal. The blood of the old animal flows into the young. This procedure, which is deemed by some to be grotesque, turns out you can extend the lifespan of the old animal by as much as 15%, suggesting there are things present in the blood of the young that extend life. Then there's this molecule, rapamycin. It was discovered on Easter Island, and it's a natural product made by soil bacteria. And this molecule got developed ultimately as a drug for transplant rejection, or to prevent the rejection of transplants. It's an immunosuppressive. But one of its interesting side effects is that, properly dosed, animals can live as much as 30% longer. You may ask, why aren't we all taking this? Well, quick answer is that you wouldn't want to. It's got a bunch of untoward side effects that no one would want to have. But the key message is that a single molecule dosed can change profoundly the lifespan of a mammal, if only you could figure out how to do it safely. So what have we learned from looking at all of these animals and these experiments? Well, I'd say a few key principles. The first is that very similar creatures can have very different lifespans. Secondly, tiny biological changes, single gene changes, can give rise to big lifespan changes. And last, that there are multiple distinct mechanisms that seem to regulate how long we live. Things like you saw with calorie restriction, or factors that are present in young blood. So, with that as some background, I want to tell you about something very cool that my group at Unity has been working on for the last 5 years. And if you accept the premise that there are multiple aging mechanisms in operation, things like the loss of stem cells, things like direct metabolic control, like you saw with calorie restriction or with rapamycin, loss of circulating youth factors, and then there's this wealth of biology that we have just yet to discover. Well, there's this corner of aging biology that we've worked on for the last half decade called cellular senescence. Now, it's not the only reason we age. We know this is because when we perturb this biology, animals continue to age. They just age more slowly, and they age differently. But what's cool about this is it's the single most druggable biology we've seen to date for perturbing aging. And it works like this. This is you at This is you at conception. You're a single cell. So you divide ultimately about 50 times, at which point there is an emergency brake that gets pulled in every cell in your body, and cells stop dividing forever. And cells that pull this emergency brake are said to be senescent cells. Now, this is a very important emergency brake. You do not want to mess with it pharmaceutically. We know this because in mice, if we genetically perturb this system, mice are born normally, but wind up full of tumors before reaching reproductive age. So do not mess with the emergency brake. However, as we age, these cells with the pulled emergency brake begin to accumulate in our bodies. So my son, who's just turning 8, has no detectable senescent cells in his body, whereas my stepfather, who died from Alzheimer's at 87, was full of these cells. Now, before our work, no one knew if these accumulated senescent cells were good for you, bad for you, or neither. And so what my group did, collaboratively with Jan van Derzen's group at the Mayo Clinic and Judy Campisi's group at the Buck Institute, is we genetically engineered multiple strains of mice in which we could clear these senescent cells from the bodies of mice whenever we wanted. And for the first time, we were in a position to ask, what happens? Well, this is what happens. These mice are siblings. They're born within seconds of each other from the same mom, and they're genetically inbred such that they're mostly genetically identical. And to give you a sense of the age of these animals, they're about 70 years old, assuming you were a mouse. Now, if you look at this picture, which one do you want to be? So this guy on the left is blind, osteoporotic, and frail. This one on the right lives 35% longer. But I think more importantly than living longer, it is a profound extension of what we call health span. This is the duration of time in which this animal lives free of chronic diseases of aging. Things like osteoporosis, osteoarthritis, kidney disease. Imagine getting old without getting old. Now, this is the picture that I carry around on my iPhone, so when anyone ever asks me what I do, I just whip this picture out. And people immediately start to wonder, when will I get to try these? And I'll get to that just in a moment. Now, since we started this work 5 years ago, we've discovered more than 20 effects, positive effects on health span, that happen when we clear these senescent cells from the bodies of mice. And I'll just show you a few of these effects. So these are the skeletons of mice with and without senescent cells. And it allows us to examine a variety of details that you cannot see with just simply crude examination of the anatomy. So if you look at the femurs of these mice, the femur is the longest and strongest bone in all of our bodies. And you look down the central axis of this femur, what you see is this profound thickening, about a 25% thickening of the wall of the femur. So, and this is in an animal that's equivalent to an 80-year-old person. And what you see is bone that looks like it's from someone in their mid-50s. If you look at the spines of these animals, so one of the features of aging that all of us will face is the gradual disappearance of the intervertebral disc and back pain and ultimately slumping and all of the features that are such a hallmark when you see an old person walking down the street. Well, when we clear senescent cells from these animals over the arc of their lives, we see a 41% improved preservation of intervertebral disc space. Now, these mice obviously look different. When you just stare at them, you can visually pick out which ones have senescent cells and which ones don't. But we wanted to have a way to actually evaluate, do they somehow behave differently? So we put these guys in a box, and we put a camera on top of this box that recorded their motion for 15 minutes. And this is what the data looks like. You see this pink line, that's a young mouse running around in a cage, running all over the place, everything it does for 15 minutes. This is 15 minutes of an old mouse. Actually, it's not a particularly old mouse, just middle-aged. And so we wondered what would happen were we to clear senescent cells, what would happen to this behavior? And what we saw was that as you age an animal to midlife, but you clear senescent cells, you get a preservation of youthful running aroundness. Basically, they run around to midlife like a juvenile mouse. But more striking, at least for me, they're willing to go to the center of the cage. We have no idea why this is, and it's very hard to get the mouse to explain it. And when people see these data, they often wonder, well, how does this become an FDA-approved drug? And the answer is, it doesn't. This is something that we may mechanistically never understand, but it speaks to the power and gravity of the biology that we're now learning how to perturb. So this brings us to the question of, when do I get mine? And just a personal point, I suffer from osteoarthritis. I have degenerative disc disease in my back. And so every day when I wake up, these data mean a great deal to me, such that I won't suffer the same fate as my father, who's functionally unable to move because of destruction of his spine. And this picture kind of explains how we're going to be getting into people. This is an old couple with osteoarthritis, clearly of the spine and clearly of the knees and probably other joints as well. And this is a disease in which you lose the cartilage that exists between your bones in the 360 joints in your body. And this disease is the primary reason it hurts to be old. Now, standard of care are things like ibuprofen and aspirin and ultimately mechanical joint replacement, but the reality is that standard of care for this disease is acceptance. You're old, that sucks, buy a cane and deal with it. That's a terrible answer in my view. It's almost just unconscionably passive. And so we as scientists, I believe, have a moral obligation to solve this. And this is a normal cartilage, and I'll walk you through how this process works. This is what it looks like in a happy, normal knee for most people in this room. As you age and begin to get osteoarthritis, this is what happens. You lose this lubricating layer on the surface. You lose the bulk of your cartilage. You wind up in pain and with inflammatory destruction of the joint, and ultimately an inability to move around functionally. One of the things we get a lot of at the company is knees. They arrive by Federal Express 5 times a week. You may say, where do we get knees? We get them from patients undergoing knee replacement. And we're able to march across the knee from where you have disease to where you don't. And we actually see senescent cells accumulating at sites within patients where you have disease. And in mice, we're able to induce osteoarthritis by cutting a ligament in the knee and inducing osteoarthritis and then clearing senescent cells and asking what happens. And we ask a mouse if it hurts. We put the mouse in a special box in which it puts 1 foot on one scale, 1 foot on another, and when it hurts, it goes like this. And these data here show that a mouse in which we've cut the ACL but cleared senescent cells are pain-free. Essentially, we've been able to cure the pain of osteoarthritis. Data I won't show you today is we can also do this in tissue taken out of human knees. We're able to regrow cartilage. We're going to be in humans, all things staying to plan, within 18 months. So, where are we? I often get this question in terms of our understanding of aging biology. The most useful metaphor I can think of is this. Pretend you're a scientist and you study a tree. Well, you could be studying the leaves. You could be studying something like the trunk. You could be studying something as pivotal as the roots. And we as scientists who work on these different aging mechanisms don't know, are we studying leaves or studying the trunk or studying the roots? But 50 years from now, we're going to have a complete picture of this tree and hopefully a set of drugs that act on each of these mechanisms. And it's an honor to be able to work on biology at this time that not only do we get to benefit from, but my son's going to grow up in a world in which a bunch of these diseases are things you only read about in books. Thank you very much. Thanks.